Roger Lewis. Sometimes when one introduces keynote speakers, there's an amusing anecdote or something like that. But Roger's too big of a deal. Frankly, he's somewhat intimidating in the list of accolades he has. So I won't be making fun of him. But I will comment that as part of our executive committee, he has sharp, incisive, and valuable comments into what we do, always delivered with collegiality and niceness. So that's good stuff to say about him. So other good stuff is the senior medical scientist with Berry Consultants. He's professor of emergency medicine at UCLA. He's an awardee of the a member of the National Academy of Medicine. That's a big deal. And he's a former chair of the Department of Emergency Medicine there at UCLA. He's had over 300 articles published talking about productivity. Senior editor at JAMA, and has been a member of the executive committee for the CTMC and has guided us since 2014. So without further ado,
thank you. Thank you very much. To put this in perspective, Megan had to catch that I had the wrong date on my slides and fix it. All right, these are my financial disclosures. I used to be a special government employee, but who knows? I am the senior statistical editor at JAMA, but this talk doesn't represent any official views of JAMA. So I want to talk about innovation in clinical trials, and I'm well aware that trainees are often at the beginning part of your career in clinical research, but I want to talk a little bit about where it's going or where there's opportunities to do things differently, and sort of try to give you a long view of it. So, you know, the pace of development of new effective therapies is incredibly slow, inefficient, and expensive. And in fact, it's particularly expensive in the United States. And that's why a lot of clinical research moves out of the United States. It's simply just a cost issue. The fact that it's slow and inefficient for many reasons, which we could talk about for literally days, impedes the developments of new treatments, and this is commonly cited as the reason why we need innovation, to make things more efficient. But that's actually not the only problem. So I want to have us sort of step back and think about the link between the clinical care that many of us deliver and clinical research. So the goal of clinical practice is hopefully to make the patient better, make each individual patient have the best possible chance of the best outcome. We also want to have a learning healthcare system where we continually learn from our experience, where mistakes, we make things better, be in a continual learning environment. There's continual change in the patient's conditions, the treatments that they're receiving. We're constantly reassessing them, especially in an acute hospital setting. They often receive sequences of therapies, and we make decisions about subsequent therapies based on their response to prior therapies. We understand that our patients are different. Every patient comes in with their own particular context and a history of their disease, and yet we still manage to make decisions about them. We're not paralyzed by the fact that we've never seen a patient like the particular patient right in front of us right then. We understand they have complex outcomes. We're trying to optimize their results across multiple dimensions. And the science that backs this up is so-called clinical science. It's some sort of derivative or flavor of what you learned in medical school and beyond. But in contrast, clinical research is different in almost all of these dimensions. The goal is to develop a product or demonstrate a treatment is effective. We in fact go to great lengths not to learn within the trial, but to avoid learning by holding everything constant, keeping ourselves blinded so we couldn't possibly learn from within the trial. We make our comparisons as simplified as possible. We often randomize at a single point and act as if everything happens at one point in time with respect to treatment decisions. We try to create homogeneous patient populations and estimate a single average treatment effect. We simplify our outcome measures, and then there's this thing for product approval called regulatory science, which is a funny thing because I don't know why we should have two sciences, but that's another long lecture. So this complete separation or near complete separation between the goals and structure of clinical care and the goals and structure of research is a problem if we want our research to actually inform clinical care. So a couple of years ago, JAMA held a summit, which is basically their name for a meeting where they invite people from the US and globally to discuss a pressing topic on clinical trials. And the conclusion of that was summarized in this manuscript, which basically says that clinical research is no longer serving the needs of clinicians. It is very unusual that when you're trying to make a decision in a clinical setting, there is a trial that tells you what the right answer is in that setting. There's trials that sort of orbit around the question, but they don't tell you for a patient with that history and those comorbidities and those concomitant medications whether a particular treatment is effective or not, in most cases. And by most, I mean the majority, but certainly not 98%. And in fact, there are a number of medical societies have looked at guidelines within their specialties and lamented the small fraction of our guidelines that are actually supported by class 1 evidence. So there's multiple ways in which this connection between clinical care and research is broken, or we talked about there being a house divided. And these are just some areas in which we need to work to repair that connection. And these are areas for possible innovation. So one is simply integrating research into clinical care. So clinical care happens with the broad population in the settings in which clinical care is delivered. Customizing our informed consent processes to be matched to the risks associated with being involved in research. So if, for example, you're admitted to a hospital and a comparative effectiveness study is being done between two commonly used treatments that are used all over the country, do you really need a 28-page consent form to approve getting a medication you might have gotten anyway, and the only difference is we're going to collect data and try to learn something from your experience? The definitions and validation of patient-centered endpoints as opposed to endpoints that are for perhaps of regulatory or historical significance. Reducing the burden of data collection. One of the things that we have to work towards is making it so we collect the variables as automatically and cost effectively as possible that we need, and not everything we can think of that happens to the patient, because I'll tell you that burdensomeness of data collection is crushing. There are obviously trial design and statistical issues to make trials more efficient and more like clinical care with learning. We'll talk about that, and regulatory science. But in all of these areas of innovation, there is a pattern that's followed. And I'm just curious, how many people have heard of the Gartner hype curve? Oh, I love this. So the Gartner hype curve is a concept that often describes the trajectory of hype, which is not productivity. Hype is the perception of value of a new technological innovation. And you can think of this as a disruptive technology, or it can be more incremental. And the idea is that early on, there's a set of risk-tolerant early adopters who jump on the so-called bandwagon. And they think this new technology is great, and they apply it in lots of settings. They have some successes. They're not that worried about the failures because they're so excited about the technology. I'll tell you, clinically, I work in emergency medicine. I think it's about the most fad-driven, social media-driven specialty I can think of. Suddenly, all my residents are doing something different, and I ask them why, and they point to a social media text, okay? So they're the early adopters. But then, the natural course is that people who are very enthusiastic do tend to overestimate the value of something. They tend to apply the new technology in areas in which its value is not outweighed by its downsides, and there is a series of disappointments which lead to disillusionment. And after that, the enthusiasm for the new technology follows into the trough of disillusionment. And there's some sort of reckoning where we all come to realize where the new technology or new approach really has value that can be sustained and is reliable. And we learn to use it in the right places and not the wrong places, and then we have some sort of stable and reasonable return on the investment in that technology. The idea here is not that everything follows this curve. This has been criticized for the fact that some technologies don't actually seem to have a trough of disillusionment, but it's sometimes useful as you look in your fields at new things that are coming on to think about where is it in this process. So one of the things that we want to repair in the connection between clinical care and research is the embedding of research into the process of clinical care, so there's not as much of a separation. An article written back, I believe in 2015, by Derek Angus, that first talk about the embedding of randomized clinical research within the provision of care. And the vision here is that you're admitted to a hospital, and when the order set is printed out for your disease, think of a preoperative order set, for example, for a routine surgery. There are areas that are the institution standard, and there are things that we know we really don't know. What is the optimal, DVT prophylaxis after a hip surgery, or what, those sorts of similar things. And those parts are randomized, where your order set might not be the same as someone else's order set, but everything you get is standard of care that a reasonable, well-trained, and competent physician might prescribe for you. And the idea is that by replacing random care with randomized care, we can actually create a learning healthcare system. Because when everybody does what they want and everything is confounded by all sorts of other covariates or characteristics of the provider and the patient, we don't learn. And this idea that big data will allow us to sort everything out isn't in fact true. But if we are just allow ourselves to randomize those things that are truly random in what we do, we have the possibility of learning. But if we do that, we have to rethink what is an appropriate ethical framework and a consent process for being a participant in research where you may not have had the opportunity to read an exhaustive consent form and partake in a long consent process. So this is an article just published this year by Nancy Cass and others talking about the right sizing of consent processes. And I urge all of you to become actively involved in discussions at your institution and beyond regarding how we can make it so that the consent process isn't a barrier that actually prevents people from participating in research, and therefore preventing them and people like them from benefiting from the long-term benefits of being included in research. A third area in which there's opportunity for innovation is how we think about endpoints. So traditionally, we're trying to accomplish, a particular sort of primary goal with a treatment. And for example, in oncology, we might be interested in length of survival or that sort of thing. And we tend to take a sort of myopic view of that endpoint. So if I'm an oncologist, and I'm trying to prevent people from dying from a malignancy, I may only look at when did they die, and not what their quality of life was, what their side effect burden was, what the effect of their disease was on themselves and others on their way to their final endpoint, if you will. But so many other things are important to patients and their families. So a utility function is a way of quantitatively combining things that are very different from each other to yield a measure of the overall value of the patient's outcome to the patient or to their family. It generally involves a trade-off between things we're trying to accomplish, like survival time, and things we're trying to avoid, like a side effect profile. And by integrating both positive and negative things, we can try to create a measure that is a better measure of the value of the treatment to the patient population that we're trying to treat. Amazingly, although it's difficult to define a particular utility function that we will all agree on, that reflects my values and your values or your family's values, what we do now commonly is use utility functions that we know no one would agree with if they actually understood what we're doing, and that's what we're going to talk about. So I want to make a comment about fruit. So when we're talking about, for example, weighing benefits and side effects of of treatments that we're investigating. A common comment is you can't really compare them and put them on the same scale because they're apples and oranges, as if it's impossible to put apples and oranges on the same scale. So I suggest to you that next time you're in a grocery store, you go to the fruit section and you see how big is the crowd of people who are just stuck at the apples and oranges and cannot decide. Okay? Okay? So for clinical investigators, the apple-oranges problem seems to be absolutely paralyzing, but for everybody else, they just look at the apples, the oranges, they make a judgment, and they decide. And the reason they do that is because we put them on a common scale called price, and you get to decide which has the best value for you against that common price. Okay? So let's talk about the modified Rankin scale. So I assume everybody's familiar with the modified Rankin. 0 is really good, 6 is dead, and it's generally accepted that for each of the ordinal levels between those two extremes, the differences are clinically important. You would rather be in a 2 than a 3, rather be a 3 than a 4, and in a second we'll get to 5 versus 6. When we're analyzing this score, which is a somewhat, or I would say a medium-rich measure of long-term disability, it is very common to dichotomize the analysis, putting everybody in 0 to 1 or 0 to 2 in one category and 3 to 6 or 2 to 6 in the other, in another category. And what I really like is by labeling the better category functional independence, we make it sound like it makes sense, right? Because functional independence sounds really important, lacking functional independence sounds bad, so clearly those are two distinctions, the two important categories. But what that means is that 3 and 6, meaning having some disability, needing some help with everyday activities, is the same value to the patient as being dead. And most of us don't believe that's true. So this is an example where a common practice creates a utility function that is wrong for everybody. If you ever see anybody dichotomize the MRS, just remember me saying that's the only absolutely wrong answer. Okay, so let's consider a clinical trial. And the MRS is the outcome. We agree that the different levels actually matter. They're important to patients. And so the question is, how do you think about treatment effects at different ends of the scale? So this is the actual diagram from the SWIFT trial. This was an endovascular therapy trial. And I want you to look at the dashed lines. So to the left are really good outcomes. So those are 0s. The right are the deaths. Those are the 6s. And the fact that the lines all slope in the same direction actually tells you no matter where you put the cut point between good and bad, these data are going to tell you the same thing. It is better to have the retriever used for your clot than not. This is really simple. It's what I might call a unidirectional treatment effect. So this is simple. It doesn't matter where you dichotomize, you should get the same answer. But what if you have something like this, where the treatment, which is I'm going to assume is the lower bar, gives you more good outcomes, but also gives you more deaths or more severe disability outcomes? How do you think about this? The first question is, do you think this is a common scenario? Okay? All right. So I use surgery as my example here. How many times have you heard in medical school or participated in discussions in whatever setting where the discussion is, if you undergo this surgery, there's a risk of death, but if you survive, this is what we hope for, okay? There's a lot of major surgeries for which that's a pertinent discussion. So that's what this is. That's the pattern you expect for a risky procedure. It can be a bone marrow transplant. You can all think of, all kinds of examples where if you get through it, the hope is that you will have a better outcome than if you didn't undergo it, but your chance of surviving is less with it than without it. And yet people make rational decisions to undergo major surgery, thoracic aortic root reconstructions all the time, because we actually do care about both ends of the spectrum here. So here's another example. Here's the TREVO-2 study. I believe this was 2 different types of endovascular devices. Someone keep me honest here, I'm definitely not a neurologist. And this is exactly what you saw, where one of the devices seemed to be associated with an increase in mortality and at the same time an increase in the best possible neurologic outcomes. And there are many people who would look at this and say, I want the device at the bottom. There are probably some people who would look at this and say that they would like the device at the top. It depends on your personal values. Okay, this is the most positive cut point, so if you happen to select that, the device looks really good, but that isn't a good summary of the entire experience. It's not very patient-centered, and then obviously there's evidence of harm at the other extreme. So what do we do? Because we need to think about ways of analyzing our endpoints in pre-specified ways that capture what we're trying to accomplish for our patients, namely measure the value of a choice of therapy for the patients in general. So this was a paper written quite a few years ago now, where we looked at the values of the different outcomes of the modified Rankin scale, looking both at how patients rated the possible states and how clinicians and caregivers rated the states. And the utility function is as follows. By definition, a normal neurologic status has a utility of 1. Being dead, by definition, has a utility of 0. So that anchors the 2 ends. And the blue and the red lines are 2 completely different approaches to measuring the value or utility. And look how amazingly close to each other they are, okay? We all really want to be a 1, a 0. A 1 isn't too bad, 2 is getting worse, and no one wants to be a 5 and 6. In fact, some people think a persistent vegetative state is worse than death. They're amazingly consistent. So if you average that, you get a utility function that looks like this. And this is actually a very good population-based measure of the value of an outcome distribution to a population of patients with neurologic injury, for example, stroke. And I think the fact that 5 and 6 are the same is something that we should remember. The analysis of this kind of endpoint is relatively straightforward. The critique is that it's not validated. It's actually much more valid than what we usually do, which I'll get to in a second, because it's actually based on patient values of the MRS. And in fact, the outcome is the MRS scale. This is simply a way of valuing the different levels of the MRS. There's sometimes the criticism that the utilities vary across patients. Some patients would much rather be alive in a persistent vegetative state than dead. Others feel the opposite. But the alternative, the thing we actually have been doing for years, and in some settings still do, is a utility function like this. You get 10 points if you're in the functional independence groups. You get 0 for everything else. And I would submit to you that is just patently wrong. Nobody has that utility function. And we should stop doing things that are obviously wrong for everyone. So my point here is think carefully about your endpoints. Try to think about if it's feasible to integrate safety and efficacy into the same endpoint. And if you do, you'll have a better measure of what really matters to patients. So this is a paper from very recently, I think 2024, on data infrastructure for clinical trials. I suggest you take a look at it. This is sort of making an argument that we need to place much greater emphasis on automatic data collection, for example, taking things out of the EHR, using wearable sensors, using those kinds of easily available but indirect measures of typical endpoints to decrease the burden and cost of data collection. This slide here is just to remind me to say that there's a lot of ongoing work in this area, and I think in the next 10 years, this will become so much more common, and the budgets will require that these kinds of efficiencies be built into the trials. Now we'll talk about what's near and dear to my heart, which has to do with statistical innovations. And this is coming back to this concept of whether we want to learn or we want to avoid learning as we're conducting research. So as you've all learned through the first part of this course, When you design a clinical trial, you don't know anywhere near enough about anything to design that trial. And your mentors are asking questions like, well, how big is the treatment effect going to be? How quickly can you enroll? What's the variability of the endpoint? As if you know. And so what you do is what any good clinical researcher does is you make guesses. And then we design a trial that will work very well if you get all of those guesses right. The problem with that is that you won't get them all right. But the opportunity is that when data start to accrue within that trial, we will learn things about the answers to those very questions, like the distribution of the outcome, the magnitude of the likely treatment effect, and therefore what the sample size should have been, that can make us make the trial better. So traditionally, we try to take those guesses and design a fixed trial where we hold everything constant, run the trial blind, and at the end we take a look and see how far off we were. In an adaptive trial, we take advantage of that incoming stream of data and change things like the sample size or other characteristics like the randomization ratio in response to those accumulating data, but according to pre-specified rules. So it's a fully pre-specified design in order to achieve some kind of efficiency. The simplest version of this is just a group sequential trial, which I think I'm going to talk about tomorrow, where you just do an interim analysis because you didn't know what the right sample size would be, and if the effect size happens to be larger than you expected, or maybe it's what you hoped for, you get to stop early because you should have designed a smaller trial. That's a very simple example of an adaptive design. The goal here is to create a learning healthcare system within the trial, where it actually learns within the trial. And I will tell you that when participants or folks considering participating in clinical trials are asked how they think clinical research works, many of them assume we look at the data and learn all the time. Because why wouldn't we be learning all the time, trying to treat them as well as possible given their condition? And they're quite surprised that the traditional approach is to enroll 500 patients and know no more when you enroll the 500th one as you did when you enrolled the first one. So there's this connection between an adaptive design and the creation of a learning healthcare system. The basic structure in adaptive design is you begin with you begin without any available data with a very traditional approach, fixed one-to-one randomization. You enroll some you enroll some patients, acquire some data, then you analyze those according to your pre-specified rules and see if any of the features of the design need to be adapted in these pre-specified ways in order to achieve some goal like statistical efficiency or perhaps more effective treatment of patients within the trial. This process continues until you reach one of the pre-specified stopping rules or endpoints for the trial, one of which may be, for example, you've reached the maximum sample size. Now, adaptive trials can be very complicated. There's a whole set of different kinds of adaptive things you can do. So there's no such thing as an adaptive, there's not like one adaptive design, it's not an off-the-shelf thing, but it's a tool chest. And the art of design and this kind of innovation is to select in a parsimonious way the subset of adaptations or complications that most efficiently address whatever the threat is to your trial. And it could be a threat to the success of the trial in definitively answering the question, or it could be a threat to the safety or the outcomes of the participants within the trial. Back in 2012, Will Moyer led the writing of this viewpoint, which simply pointed out that when we use one adaptation, which is response adaptive randomization, where you tilt the randomization towards the arms that are performing better, that at least partially addresses this disconnect where patients or participants have the therapeutic misconception. They think the trial is there to help them, when in fact, most trials are designed to not help them over time. The next extension of an adaptive design is a platform trial, which is a trial that's intended to survive the evaluation of any individual therapy and essentially have a sequence of therapies that sort of come into the platform, get evaluated, and then may leave the platform either when they are successful or if they fail. The efficiency of a platform trial is that as you all are learning or have learned, the process of starting or closing out a trial of any sort is very burdensome, time-consuming, and in some cases expensive with respect to personnel. So if instead of creating and then breaking down a trial, every time we want to answer a clinical question about the optimal treatment of a disease, but we always have the trial running and it's the therapies that drop in and out, we can save the inefficiency associated with startup and shutting down. Now, platform trials can be very complicated. You could start, for example, with a control in 3 arms. You could have some other arms drop in. You could have an arm drop for the finding of futility or harm. You could find that one of your treatments is superior to the control. And then that treatment can actually become the new control. So I want you to think for just a second, if you're doing a multi-arm trial with a standard of care control, and one of the actives is better than control, for most trials you're done. Your control is no longer an acceptable alternative because you've shown there's something better. In a platform trial, those sorts of changes can be accommodated, although I admit it is not non-trivial. This is a commonly cited article from New England Journal back in 2017, written by 2 personnel at the FDA at the time, sort of outlining this approach and its appropriateness for the development of evidence to support regulatory approvals. So why do we care about platform trials? Why is it an important innovation? One is simply to address that inefficiency that I mentioned at the very beginning, that clinical research, it costs too much and is too slow with respect to answering each individual question. So we can have efficiencies related to a shared control group. If you have 3 drugs versus a control, you don't need as many control patients as if you did 3 separate trials, each with its full control group. You avoid the costs of the startup and the closeout. There are some statistical efficiencies that have to do with the randomization proportion or the stopping rules or enriching the design, enriching the trial, which means starting with a broad patient population and then learning what is the subset of that population for which the treatment appears to have the greatest benefit, and then limiting future enrollment to that subpopulation. And then there's a set of other statistical efficiencies that are most commonly used in this setting that can further speed the answering of questions within the trial. So where are we in the use of these kinds of approaches, and this particular one, platform trial approaches within medicine? Well, it turns out that medicine is not a single field, and that there's different rates of uptake in different fields. And I just did this based on my own experience and impression. This is not a scientific graph, just to be clear. Adaptive trials and platform trials started first in oncology. There's most experience with that. I would say oncologists are the most familiar as a medical readership of interpreting results from trials like this. There's other areas like pediatrics and women's health that are just starting to show some interest in these techniques, and there's the enthusiasm that comes with being on the early part of the hype curve without the pain that occurs when you find out you've fallen off the top. Critical care, there's been a lot of activity in this during COVID. There are a lot of results from platform trials in COVID. Neurology is an interesting story. So in neurology, there have been a number of successful implementations. For example, there's the Healy ALS platform trial. How many people are familiar with this trial? Okay, I should not be talking about this. Recently published in the recent time has published a series of results. And the point here is that, unfortunately, the results have been disappointing from a clinical benefit point of view, but from an efficiency of getting answers to as many questions as possible so the field can move on to whatever the next most promising agent is, that it has been very effective. Here's an example from Lancet. Here's one from JAMA. Here's another one from JAMA. Here's one from JAMA Neurology, and one from JAMA Network Open. These are all within the last year. Okay? So the platform is doing what it's intended to do, which is continue to be of value beyond the evaluation of a single intervention. What about stroke? So this is a review article that Liz Lorenzi and others recently published on stroke and the use of platform trials in this setting. This is something that's near and dear to our heart. But before we talk about it, and in the closing, I want to talk about the tragedy of the commons. So how many people know what the tragedy of the commons is? Okay. That's better than talking about the healing platform trial. Okay. So what is a tragedy? I had to look this up. So it's a drama, and the thing that marks a tragedy is that there's some sort of conflict or problem, and there is an irresistible force that will ultimately lead to a disastrous conclusion. It's a very upbeat kind of drama. Okay. What is a commons? So a commons is a piece of land. Notice this is the fifth definition. I'm being somewhat selective here. A piece of land subject to common use, especially used for pasture. So there's an issue with the sharing of pasture space that was first described back in the 19th century, but a population biologist by the name of Garrett Hardin placed this into a more modern context in 1968. And if you want to read about an interesting guy, Garrett Hardin is a very interesting guy, but here's the idea. So you have this shared pasture space that is a resource for everyone. And the rule is that everybody is allowed to graze their sheep on this shared commons. And this is really appealing. It's simple and it's clearly equitable. Everybody has access to the commons. But each herdsman, and I'm going to go out on a limb and say in the 19th century they were men, each herdsman seeks to improve their situation by deciding how many sheep to graze, right? You can decide, how many animals to bring each year. But the land has a fixed carrying capacity. You can't make the commons bigger. So what happens is that from the point of view of each individual sheep herder, if they add more animals to their flock, they get almost the benefit of one additional or two additional animals. That animal is completely theirs. And the net effect of overgrazing from that one animal or two animals is distributed among everybody, so the benefit accrues to you as an individual, but the harm is distributed. So the rational decision for every herder is to add animals to their flock. And ultimately what happens, and this is the tragedy, is that the land is overgrazed, the flocks do not survive, and everybody starves and dies, so it's a tragedy. Okay. But the idea here is that if you allow free access to a common resource, common limited resource, that's actually not good for anybody. So I want to bring back this idea back to clinical trials. So there is a bunch of commons-like resources that are important for clinical trials that we need to figure out how to manage. So there aren't that many productive sites and teams, and we all know that every stroke trial is trying to get the same highly productive sites to participate. The number of research, especially for some of the diseases you guys are investigating, the number of participants is very limiting. The number of treatments that are promising to test. And certainly the funding, and I don't want to spend a lot of time on that, but there's no question that the funding is a limited resource. So in many fields, there are lots of either commercial sponsors or investigators or teams of investigators all trying to use the same limited shared resources, basically get access to the commons. And the problem is that if we don't manage this among ourselves, we can end up with all of the efforts or many of the efforts failing because the sites, for example, are distributed among too many teams and no one is getting the enrollment numbers they need in order to answer the question. So how do we avoid the tragedy of the commons? So in other settings, we simply regulate ourselves. We decide that the commons is not shared, that we have private property, or we have regulations on the use of shared resources. Think of national parks. So the idea is they call about mutual coercion, mutually agreed upon. The idea is we agree for the good of the shared resource, we're going to limit people's access to it. And there's examples in the healthcare system that illustrate this. So how does this apply to platform trials and stroke? Now, when I made this slide about NINDS, I was going to give this talk at a place where there was no personnel from NINDS. So what I'm about to do, I would not recommend any junior investigators doing. Okay. So after the demonstration of the incredible efficacy of clot retrieval for large vessel occlusion, acute ischemic stroke, it was seen and I think anticipated that there was going to be a large number of investigators who wanted to figure out how to expand the indication. What are the limits of the population? Think in terms of the stroke characteristic, the population itself, the timing, or the limits in terms of other concomitant therapies that would increase the number of people who could benefit from this kind of intervention. And yet, if all of those grants went in as single grants, single projects, each one with its full control group, set up costs, set of sites to NINDS, and they tried to review all of those and fund all of those, it wasn't going to be the best use of our shared public dollars. And this is a slide, it's not my slide, I pulled it from something else, that just talked back around 2018 and 2019, what were the status of multiple efforts to develop clinical trials that were ultimately going to be submitted to federal partners for investigation of therapies around endovascular therapy. And the idea is this was going to create a commons problem, if you will, where the NINDS's funds was the commons. So this is my interpretation of NINDS strategy. I want to say I have no inside knowledge of this. What they did was they published a notice of special interest that basically said, we think we need a platform trial in acute ischemic stroke. Do we need to create a managed commons, my term, not theirs, that we can then use to most efficiently and in a cost-effective way evaluate as many of these important clinical questions as possible. So first there was a notice of special interest. A lot of stuff happened around that period of time. And the mechanism is, as we've mentioned earlier today, an OTA funding mechanism. It's a two-stage mechanism, which was also described earlier, where the investigators come in with an idea, they enter a collaborative design process, and then there's a second stage competitive application process to decide what actually is tested within the platform. I'm just going to pause there for a second. Was that an okay summary?
Yeah. I mean, I could give a little background. If you want to.
Yeah, I think this would be good. It is very rare you get to hear about this from the decision makers.
I can maybe have a microphone.
Yep.
So as Roger suggested, there was a lot going on at that time, and what he's referring to is the fact that the DAWN trial, well, the 2015 thrombectomy trials had basically shown that clot retrieval was beneficial, and then in 2018 we had the DAWN and the DIFFUSE trials, and both of those showed that you could do thrombectomy up to 16 or 24 hours essentially. And we funded the DIFFUSE 3 trial, so immediately when that was reported at ISC, the International Stroke Conference, and even prior to that, based on the investigators involved in DIFFUSE 3, we had a lot of questions that were around that, like what about folks that are older? What about kids? What about a little bit of a longer time period? Or what about infarcts that are a little bit larger, large core infarcts? And just all sorts of questions started coming at us. And actually, I realized at that time that we really weren't going to be able to answer that sequentially with individual trials. And Walter actually had the idea that we should really not only understand, what's beneficial, but also what is not going to work. And so, I had been interested in platform trials, and so we concocted this idea of doing a platform trial in EBT, and that's what this was based on. And so I'll give it back to you. But the NOC essentially was, we were interested in this, and we'd never done it before. And there was no way to do it with traditional grant mechanisms. I mean, you cannot have multiple trials coming in through separate applications. How would you interdigitate them? And they'd all be on different timelines and all of that. So really this OT made it possible for us to do it. And I don't know the answer to this. Maybe Adam knows. Some of the other trials that have been done in NIH, like at NCI, I don't know if they used other transactions or not, like the iSpy trial or the GBM Agile or whatever one mechanism. Yeah, well, they have those cooperative groups though, which make it a little easier. Anyway, that's the story.
So, and I just want you to know I'm not seeking funding from NINDS. I will say that I think it's a remarkable show of intestinal fortitude to do this, because it required a period of time where they were not reviewing submissions in this area while they put this strategy together. And I suspect that made some people unhappy. I don't know that, but I can't believe how it didn't. So there's the STEP platform trial has 38 sites and some Canadian sites, and is actively enrolling. And so this has actually come to fruition. So, in conclusion, even if you're at the beginning of your career in clinical research, I think you want to think about innovation and where there's opportunities to do it better going forward than the way we've done it, over the last decade, 2 decades, 3 decades. Innovation is not accidental. It's based on understanding where there are limitations in what we're doing. And I think thinking carefully about your dual lives, for those of you who are clinical investigators, between the way you provide care and the way you investigate the efficacy of treatments, and trying to figure out where there's opportunities to make them more relevant to each other are important areas. The trajectory of innovation is not monotonic. I think the hype curve is a useful conceptual model for at least thinking about where various things are. We need those early adopters, and we need the people who think critically about those early adopters' activities to get us to the right place. Most of the examples that I've used today are based on large, multidisciplinary teams. I think one of the things that's very daunting when you're early in your career is seeing these things that are done by teams of 30 or 40 people. I would suggest to you that while you're working on your own individual research activities, which are important to your career trajectories and promotions and things like that, that if you have an opportunity to join a large collaborative effort, you do that as well, because there are opportunities for mentorship and developing leadership and learning how things get done that will be invaluable whether you stay involved in large team science or not. Pay attention to innovations in other fields. There's actually nothing special about the brain with respect to how clinical research is done, I'm sorry to tell you. But you can look really brilliant when you bring an idea from another field into your field and people think you thought of it yourself. And take advantage of all networking opportunities you can. Thank you very much for indulging me, and have a good evening. Happy to take questions if it's okay with Mark. Okay.
The utility weighted modified Rankin score.
Yes.
Brilliant. Do you think it's different for each disease? Because it's specific to that patient population and their perception of what their quality metrics are?
Sure. So the question is the utility function different for each disease? So yes, but not that different. One of my favorite phrases, and I apologize to people who have to hear this from me, is don't let the perfect be the enemy of the good. So the utility weighted MRS, which is based on particular populations, is a not unreasonable measure of the value of the different levels of the Rankin. You can use the Rankin as outcome after head trauma, after stroke, after other neurologic insults, even though it wasn't developed for those. And it's not a bad measure. It's much better than trying to dichotomize into a good versus poor neurologic outcome and giving those utilities of 1 and 0. So I think it's better. But there may be other settings where we need to spend the time to develop utility-weighted approaches to other ordinal outcomes. But in the absence of that, starting with this one is not bad. If the now remember, that relies on the fact that the modified Rankin is a measure of disability for your disease in question. Okay. Other questions? Anything else? Well,
But when you use it that way, there are like so many zeros. That doesn't seem like a normally distributed outcome anymore.
do you want to answer your own question, or do you want me to? Yeah. So so I'm glad Kurt left, actually. So the point that I believe Will is making is that even though it's not a normally distributed outcome, that for any reasonable population, there's a theorem, the central limit theorem, that says you get a pretty much normally distributed distribution of the average outcome of the two populations. And so most of your statistical stuff actually works just fine with the t-test. I will tell you that we've recently had some experience where we've been simulating this in detail, and the approximation isn't as good as I thought it was, and so it does sometimes take some work to make sure that your analysis was right. But the advantage of the utility weight over a simple ordinal outcome, ordinal analysis like a Wilcoxon rank sum, is it keeps it pays attention to the distance between the values of the adjacent categories, as opposed to a Wilcoxon rank sum, which only keeps track of the ordering, and that gives you a little more power.
And I guess, how many statisticians are still in here? A lot. Oh, but you're here. You'll be. A lot of times, in a situation like that, you may get well-meaning advice from statisticians to use some form of a non-parametric test. And again, as you point out, in that you're changing your assumptions. All of these assumptions have problems if you're violating them some in every sort of model-based approach. And there's often a bias towards, well, this is a little more conservative to use a non-parametric approach. But unless you simulate it, you don't really know what you're really trading on.
Right, what the trade-offs are, absolutely. Anything else? Okay, thank you very much.
Okay,